Hello guys, and welcome to my channel. So I am a teacher, a biology teacher to be precise. So today we are going to talk about food test. Yes, you heard that right. So these are my food samples and my reagents. Yes, these are all of them already prepared. So let's go into the actual video. Zooming. Yep. Okay, so the first test I carried out here is the starch test. Yes, the test for starch. Yes. So the first thing I did was to add my starch sample, after which I went ahead to add my iodine test. Yes, iodine test is the test for starch. So I go in with my dropper and I picked a reasonable amount, and then I added few drops to the sample in the test tube. So you can already see a change in color. So I have to shake a bit for them to, yes, that is it. A blue-black color indicates the presence of starch. Yep. Okay, so the second food test is the test for sugar. Yes, the test for what? Sugar. So I have to, and by sugar I mean glucose. So I had to go in with my glucose sample and then my reagent. So for this I'm using a Benedict solution. So this is me adding a drop to the food sample, after which I try to boil. Yes, you have to boil in a hot water bath. So pardon me, I'm using a frying pan. So you have to boil for at least 3 to 5 minutes, depending on how hot your water is. So I started from the scratch here. So this is us boiling to expect a color change. So we'll keep doing this until there is a color change. Now a color change indicates that glucose is present. So let's watch how the color transcends from what it is now to what we are expecting. So you can see it has changed a bit, but this is not the end product. So we keep boiling. So if you don't know what the end color is, you may want to pull out now. So I just experienced it. No, we don't do that. So we have to keep boiling, and you see the color keeps changing. You see now it has changed again. Now I think this is now lemon green. I'm not very good with colors, so this is it. So we keep boiling. We are expecting a particular color change, so we will not stop until we get there. You can see this, it's already moving again. Okay, and now we've gotten at least, I think this is yellow, if I'm not mistaken. Yes, yellow. So a yellow precipitate or yellow color change shows that glucose is present. Yes, a yellow, or for some people they can hit till orange, so it's almost tantamount. So this is it. Voila. So this color change indicates that glucose is present, and then we put back in our test tube rack and then get ready for another food test. So the third food test we're doing is the reducing sugar. Yep, you have that right, reducing sugar. So we're testing for sucrose basically. So we add our food sample and then we go in with the same Benedict solution. Yes, so after which we go ahead to mix up, just shake thoroughly, and then we go ahead to heat like before. Okay, here you can see that I still added more because I wasn't convinced that that drop I added was enough. So you see I go in with my test tube holder, properly fitted, and then back to the hot bath. So this is it. Yep. So we boil for a couple of minutes. Yes, we boil for a couple of minutes to check if there will be a color change. Okay, let me tip you off. Unfortunately, there will be no color change because we have not carried out hydrolysis. So you see no color change when we just use only Benedict solution in our reducing sugar. So the next thing I did was to go ahead and get another test tube and then the same food sample, which is the reducing sugars, and then I go in again with my, now this is what we call hydrolysis now. So I go in again with my hydrochloric acid first. Yes, hydrochloric acid first before the Benedict solution. So when we talk about hydrolysis, now let me just walk us through what that term is all about. When we talk about hydrolysis, we are really literally talking about the breaking down of the reducing sugar into simple sugars. And to do that, you need your hydrochloric acid and your sodium hydroxide to be able to carry out that. So back to what I am doing here. So you see, I've added all that, just my hydrochloric acid and the Benedict solution to the food sample. So I go ahead to boil. So I boil for a while, and guys, let me tip you off again. There will be no color change. Yes, in fact, it literally turns colorless. Yes, let me use the word. The solution becomes colorless. So what do I do next? I go ahead to cool. Yes, I cool off that in my beaker filled with water. Yep, that's it. So after cooling off, just stay for a while, and yeah, voila, I'm back again. Now what do I do now? I added my Benedict solution to it, and as soon as I did that, you could see a color change, and then my sodium hydroxide. Yep. So my sodium hydroxide. Yes. So I've added my sodium hydroxide earlier. Sorry for that mistake. And then this is my Benedict solution. So this is actually my first time recording. So I hope I did a perfect job. So you can see the color here already. So we go ahead and we hit, and guys, let me tip you off again. This color change was almost immediately, like rapid. So you could see it became more blue in color, and then it begins to move. Just watch that. Can you see that? Wow, this is magic. Oh, well, it's science. So this is it. See, rapidly just moving just from 0 to 100. And oh, wow. So we are seeing a different color change, but we are not stopping here. So, yep, this is what we have. This is actually orange. From orange now. Oh, it's becoming thicker. And oh, wow. So this shows that reducing sugar is present. Yes, sucrose is present basically. So yep, this is it. So you may be wondering, oh, food test. My dear, this is what we do in our lab. Okay, so we are done with this. So we have to place it in the test tube rack. Yep. So we move on to protein. Voila. So this is protein, and I am adding my food sample to the test tube. So after which I go in with my reagent. So for this, I'm using Millions reagents. Yes, you heard that right. There are different reagents, two major types of reagents when testing for protein. Either you use your Burette test or Millions reagents. So I'm using Millions reagents. So I go in and add a few drops. Yep, so you can see that I'm shaking that thoroughly, and then I go with my test tube holder. And again, guys, I go ahead to boil gently. Yep. So I will be boiling this for a couple of minutes, yes, because we are expecting a color change. Yep, expecting a color change. That's it. So you can see we're boiling for Okay. Yep. So guys, like I said, this is my first time. Make sure that when you're watching this video, you drop a comment, whatever you want me to do next, like an encouragement. You can even share, like, subscribe. I'm a teacher, a singer, like I'm basically so many things, even a social media manager and a content creator. Okay, back to our video. So yes, you can see the color change. It has moved from the white precipitate to this now. Yep, but this is not what we want. We are expecting a brick red precipitate, so we will be heating up again, and you can see the color change. By the way, my name is Desire. Yes, Mr. Desire. Most of my students fondly calls me Mr. Teacher. So yep, that's a brand I'm hoping to uphold in future. So yes, this is what we have. Keep hitting. As a biologist, there are some things that when I read in textbook, I'm like, I need to see this practical happen in real life. I need to, back in the days, I didn't really have so many opportunities to try out what I read in my textbook. But being a teacher, when it's time for practical, I'm even more excited than my students. Yep, you heard that right. So this is it. It has changed. A red brick precipitate. Yep. So this shows that protein is present. So basically, the food test is just used to analyze the type of nutrient in food. That's it. Like the kind of nutrients in food. Is it starch? Is it fat? Is it protein? Is it sugar? Is it, and all of that. So most times, if there are any food that you are in between, okay, am I good? Okay, so sorry, let me come back to what I was doing. So the next one is the test for fats. Yes, so for this I added my food sample. Yes, should I tell you what it is? Okay, let me try. That's just a peanut butter. Yes, so I go in with my Sudan 3 test. That's what we use for test for fat. Sudan 3 test. You can also do the emulsion fat test and all of that. So this is it. What do? I have to just shake, and this is one of the simplest. Yes, it's just one of the simplest. It just turns to a red stained layer, showing that what fat is present. We can also do the pepper smear, like if it is oil, you can just do your pepper smear, and then it becomes translucent and all of that. So guys, this is what we have. So this is the carbohydrate, that's the starch, the blue black color, that's the protein there. Yes, that is the first one, which is the glucose test. We have the first reducing sugar that didn't change color, and then the second reducing sugar that changed color, and then we have fat. So thank you so much for watching. Make sure you hit the subscribe button, like, follow, comment, and share. Bye bye. So this is me, fine teacher.